word of SARS sort of emerged, there were studies which came out, and the first was from an Indian group, Pron et al., who published a paper called The Uncanny Similarity Between HIV and SARS-CoV-2. And they identified that there were homologous epitope sequences in the spike protein of SARS-CoV-2. And this was jumped on by from right on high, right at the beginning of the pandemic. They were forced to retract, or not even retract. It was put up as a preprint, and it was taken down. And there was a coordinated effort at the highest levels of NIH and the public institutions to squelch that finding. So the technical term would be like prion-like. I'm not sure I like prion-like. They're just prions, which means proteinaceous infectious particles, which means they're able to, like I say, start sticking together and form these cascades. We're looking at, well, okay, we're seeing fingerprints of what look like, so my friend Charles Rixey calls it de facto biowarfare. Whether it was deliberate or not, at this stage we don't know. But you just have to presume that, going by the evidence of what's emerging and what we know just about the molecular biology.
All right, welcome to Health Alliance Australia. My name is Jeannie Rose Anschweifer, and I'm here with Dr. Kevin McCann. Health Alliance Australia is a leading health advocacy organisation in Australia, gravely concerned about the weaponisation of medicine and science for political power and harm. We are dedicated to honouring bodily autonomy and well-informed health choices. We provide health information on our website, healthallianceaustralia.org. Through our podcast, you can subscribe to our newsletter, donate, or join as a member. We've also donated over $200,000 to various legal actions. Dr. McCann is an advanced neuroscience researcher. He's one of the few in the world who's discussed how COVID-19 as a pathogen is affecting the brain and the central nervous system. The same patterns is associated with different diseases that he works with in his laboratory. Through our previous guest, Dr. Richard Fleming, and bioweapons expert, Professor Francis Boyle, we have established that SARS-CoV-2 is of lab origin, a bioweapon with gain of function. Essentially, they've created airborne AIDS, and that using mRNA gene technology, they have spread this spike protein toxin from the vaxxed to the unvaxxed, and that through infection, shedding, and injection, the spike protein is all harmful, the latter being potentially the most harmful. Those interviews are available on our website, and so if you haven't seen them, please go back and watch these. But today we are planning with Kevin to discuss the potential mechanisms of harm of COVID-19 to the central nervous system, prion disease, amyloidosis, mRNA technology, and potential treatments. Because, you know, what you're sounding the alarm on, Dr. Khan, is potentially a ticking time bomb. So if I could just ask you first to give a small brief introduction about yourself, and then we can discuss what prion disease is.
Sure. So as you were saying, I'm systems neuroscientist. I specialized in, well, still a specialist, but before having to step out of the orthodox institutes doing this, I specialized in what's known as cortico-basal ganglia disorders, and I had a interest in a technique called deep brain stimulation, and where it had been developed for neurodegenerative conditions like Parkinson's disease. I was sort of working on trying to develop that for quote unquote neuropsychiatric disorders, I guess, OCD, that type of thing. And so I had to have feet in both worlds, the neurodegenerative and the neuropsychiatric. And as a consequence, I was fairly attuned to the literature as someone who worked with primates, trying to model these disorders. And I was one of the first to be publicly calling out that what we saw happen at the end of 2019 into 2020 was at minimum a consequence of something that's happened in a lab. And that was contentious in and of itself 4 years ago. And in trying to explain to people what potentially could happen with respect to exposure, I ran very squarely into what we now know is a weaponized and highly controlled information ecosystem designed towards censorship, narrative control, and behavioral control through advanced neuropsychological techniques. And so I was able to sort of pick apart some of this stuff. I would have to deal with, I've had to deal a lot with people who think viruses aren't real, and basically try and find a way through both sides, from the orthodox science to the like I said, I don't. I understand people wanting to find answers to questions. So for the lay public, I have time, but for those that have positioned themselves to exploit what happened over the last 4 years through either you're selling books that viruses aren't real, or you're selling therapies that are unproven in this instance, then I'm going to address you and follow and engage you on the internet on in this information ecosystem that they've put in place. And so that's how I sort of got into this. And as this progressed, what we find is that the authorities were ready to roll out a completely novel, as a public health measure. It wasn't a novel technology in terms of research laboratories. And this, in my mind, was an unconscionable breach of, well, bodily sovereignty and well, your right to informed consent. And it's pretty obvious from government actions that they were not being forthright with what happened. And, as they told everyone that this was a zoonotic event, etc. And they weren't being forthright about what it was that they were proposing as a intervention around the vaccines and the RNA. Now, why was I so concerned? Well, in my field, um, back in sort of 2014, 15, the scientific landscape began to change as we began to understand neurodegenerative disorders as the canonical ones: Parkinson's, Alzheimer's, these types of disorders. We began to understand them as types of prion-like disorders, meaning that there was the same mechanism, disease-like mechanism at play, which is that you have a misfolded protein that then initiates a cascade, and this cascade then causes the outward expression of the symptoms that we recognize as the neurodegenerative spectrum. And it was trying to inform people about, well, how that occurs, because the major suspect for what's termed idiopathic neurodegeneration, meaning we don't really know the cause primarily. You just turn up at the clinic. There's no real genetic markers as to why you should be having these traits. It's just called idiopathic, and like I said, it's you just classed, and depending on how you respond to therapy, will depend on how they sort of class the disease, and if they're really interested, they might make a post-mortem diagnosis. And this, trying to get across to people that there's this crossover mechanism of this protein misfolding assault that can occur, and can occur theoretically, and I didn't have the data to hand at the time, but my presumption was that if what we're dealing with is of lab origin, that it's highly likely that you would see a move in that space towards the weaponization of prion or amaloggenic, we call it, disease mechanisms. And so, and I was fortunate enough to have the only monkey in captivity to develop an early onset. What we thought of at the time, and this was around the time that the literature was changing, developed this early onset Parkinson's like condition, and had all the hallmarks of um, well, prion-like disorder in the brain, let's say. And so, again, I was sort of wrong place, wrong time from my perspective now, but there I was and trying to lead, not even lead, just speak out. And that's how I've ended up here talking with you.
Yeah, I mean, I think when we were looking back, say, to the Wuhan strain and the unnatural, what would they call it? Silent hypoxia, where people had these very, very low oxygen levels that weren't conducive to actually life. And then suddenly they were either leading to, they were having organ failure or death. It seemed right back then that might have been one of the warning signs that we were dealing with something very, very different to just a simple respiratory virus. And now when you actually look, say, at the mRNA technology that's been rolled out, and we have all this unnatural dropping dead of people with sudden heart attacks, people dying in their sleep, young people dying. There seems to be a different mechanism to what is official. Like, I mean, we've classed it as vaccine injury or vaccine death, but there seems to be something much more going on here, because it's like a switch has been turned off into people's brains. And I think this medical and scientific community aren't really looking at that aspect of it. That when you actually, I mean, plenty of people have heart attacks, and they survive. They get treated. They live many decades afterwards. It's a very new thing to have, like, sports people have heart attacks on the field and just die.
Oh, it used to happen all the time down my way. What's the matter with you?
Do you know what I mean? Like, it's yeah. Like, so we're dealing with something that is kind of like a zombie apocalypse almost. Like when you're looking at, and of course now we're trying to tie in together with what is being reported in autopsies with these white, fibrous clots. I mean, do you think these are amyloids forming or something else?
I think it's something along those lines. The literature is pretty clear now that exposure to the spike protein, it doesn't matter where from. In fact, it may be worse to push it past immune barriers, causes a reactive amyloidosis. And the most immediate presentation that is seen is this the formation of what are called fibrinogen resistant microclots, meaning that they're, as the word says, they're clots in the blood, and they're like, and so prions are categorized by being primarily, not all the time, but in most of the normal species that are encountered, as being resistant to protease digestion. And in this instance, fibrinogen. Hang on a second. You can sit there, big boy. I know there was tech problems with Miss Gale. Okay. Sorry about that. And so this was being observed relatively early on in the patients who were presenting with the condition. The South African group, Pistorius Lab, they were sort of some of the first to identify that you could get these amaloggenic occlusions in the blood just from exposure to the what we call the S1 segment of the spike protein. And what you have when you have amyloid like processes is that they basically, you think of them like Lego. They essentially just stick one end to the other till they form very large fibrils that cause a reactive inflammation. And if you have them at the level of your circulatory system, then you're going to basically go into a sort of chronic hypoxic state. This is going to compound the disease state to the point where potentially you could be pushing yourself over into if it's not if you're unfortunate enough to go past go and straight to past long COVID and straight to death, you might be passing through stages of dysautonomia that can occur, whether blood pressure and autonomic control of the body begins to fail. And this is something that people need to wrap their heads around, which is that when I'm talking about prion, even though it's famous for the neurological presentation, the impact of misfolded proteins can impact every level, organ, and tissue system of the body. And in most cases, you have this exquisitely evolved immune system and surface protection that can, would deal with this, and you have a generally, if you're lucky, a normal life. And the neurodegenerative aspect is an end of life accumulation of these aggregates that occur. And if you something we all have to pass on, otherwise there's no
I mean, I
room for
think you were talking, before we were talking about how you were saying 1 in 2,000 in the UK when they were autopsying, they found that they had the spike protein in them.
No, this was from studies into from exposure to the BSE, bovine spongiform encephalopathy prion from the 80s.
Oh, okay.
And they'd done a sampling of approximately 30,000 appendectomies. They're taking the appendix out. They would screen them and look for signs of the condition starting at that, because the appendix is part of your immune system.
So essentially, what we thought that prions were rare, I guess. It's more or less an environmental toxin, even going back to the mad cow days. That here, when Jean-Claude Perez's latest paper came out, and he was talking about how with the mRNA vaccine, that it was actually essentially shedding these, what did he call them, parasitic proteins and prions. So when people are vaccinated with a mRNA technology of some kind, they are essentially producing these spike proteins that could potentially cause other people to have amyloidosis. I mean, that's my reading of it, that they're becoming like a bioweapon, essentially.
It's a bioaccumulative, highly resistant to degradation. You can basically think of it almost like a toxin building up in the environment. What Jean-Claude Perez was actually discussing in this instance was the process of frame shifting that they have now admitted to with respect to using the pseudo-uridine as a replacement for uracil in the mRNA shots. And what that does is it causes a slip with respect to where the reading frame begins and ends. And what he showed was that you can theoretically produce a bunch of foreign peptides that are outside the scope of the what the products were that they were hoping you would translate. And it's not it's a complex picture, but the idea was that by giving you the source of the spike protein, you would make an immune response to it, and the body would deal with it as sort of immunology had sort of predicted, and it would confer immunity, and then we could all go on about our day. But because of this particular type of technology, this was work that came out of Cambridge in the UK. They showed that, oh yes, we do get frame shifting, and what you see when you look at the expression products from using the mRNAs, you see when you do something called Western blotting, which is you sort of try to draw out the proteins by molecular weight, and you would stain them on a gel and get a reference molecule that's supposed to be your product. And what you should get a very discrete band which shows that you've got the target product in this instance. And what they find is that the results are smeared out, and so there's lots of peptide fragments there that are not part of the original plan, as it were. And these off-target expression proteins could potentially be deleterious in this instance. And he showed that there was a lot of homology. I actually did an edit on his paper just to try to tidy it up, get it into a sort of more Anglophone context. And there was a number of peptides that were expressed that were, one was, it's the STD that that all the French royals used to have, and it slips me at the moment, but and one from a brain-eating amoeba that was being expressed.
Oh, yeah,
And
yes.
and so there's a lot of incongruities around what was being sold to the public and what actually was happening. And so we have to look at where we are today versus 4 years ago, right? We have to see where the data has plotted. What is it showing us and telling us? And from that initial period, we fairly certain that, we know that these biowarfare research programs were geared towards trying to find, and this is a consequence of the post 9/11 and the anthrax tax, and billions of dollars were poured into that space. And what they did is they essentially were honing down the properties of pathogens. Oh, syphilis was the one I was just trying to remember, but they're honing down the reactive sequence of peptides that come from pathogens in the environment. So take, for example, syphilis, right? There's going to be a bunch of proteins associated with it, and they will have adapted to overcome the host's immune systems. And in the case of syphilis, it's one of those diseases which does impact the central nervous system in very direct and often lethal ways. And so in this environment of this biowarfare domain, as they've looked across pathogens and said what peptide and what epitope, meaning basically you can think of 20 amino acids long, right? So it's a tiny fragment if you think that the spike protein of SARS is approximately 3,000 amino acids in length. And so 20 amino acids, that's it's small, and they this seems to be a sort of fundamental read level within your biology, because the body itself uses these short chain RNAs as signaling molecules as well. So we know that there is this coordinated biology down at that scale that the organism uses. Okay, so people need to be conscious of this type of biology when trying to understand the problem that we're dealing with. And this is going to be a very sort of roundabout way of getting to the prion hypothesis. So as word of SARS sort of emerged, there were studies which came out, and the first was from an Indian group, Pron et al., who published a paper called The Uncanny Similarity Between HIV and SARS-CoV-2. And they identified that there were homologous epitope sequences in the spike protein of SARS-CoV-2. And this was jumped on by from right on high, right at the beginning of the pandemic. They were forced to retract, or not even retract. It was put up as a preprint, and it was taken down. And there was a coordinated effort at the highest levels of NIH and the public institutions to squelch that finding. And what they tried to do is try to pass off by getting talking head, and these were well, they're scientists for hire, basically, who work within the institutes, to say, well, okay, there may be some homology with HIV, but they happen to exist. So if you look, I could bring slides up to help sort of illustrate the point, maybe.
No, if you want to, I've got the screen share available.
It might just help sort of focus the discussion. And
So are these small fragments? Is that what is leading to so they go into the cell and then the cell misfolds from there? Is that what you're saying?
yeah, so you can have these fragments and these epitopes. So we know that there's homology with HIV called GP120 and GP41, and so what they're trying to do. The screen should share, right?
I think while we're waiting for this, I'll just read a tweet that Dr. David McCartland had written. I'm pretty sure everyone's seen it recently. It says, prion disease is coming. It may take years to manifest, but it tragically will. There is a plausible mechanism, the one whereby we inject a human with an mRNA toolkit by the billion, with the whole body biodistribution and coated with liquid LNP nanoparticles for maximum transfection capacity. The toolkit for a non-human spike protein construct, one that crosses the blood-brain barrier. So, and I think he's been quite outspoken about how it's also, in his eyes, demonstrated personality changes within the medical field that he works in. And he is putting it down to the action of prions. It's quite dire.
He listens to me.
Yeah, it's quite a dire prediction, right? And I think also there's some element of a spectrum of disorders, from being asymptomatic to having mad cow disease, I guess.
And so this was a figure made by someone called, it's known on the internet as Jicky Leaks. But you can see this, right?
Yeah, I can. Yeah, it's all good.
Yeah. So what they tried to tell the people was that this sequence homology, oh, it's just essentially random and it's just mere chance that these loops, and so they're signified by yellow on this. So if you rotate it in 3D, which it should do,
Oh,
these
yes.
functional loops are, it's just chance that they have this overlap. But what they didn't tell you is that this overlap comports with the with three different clades of HIV and of clades across different countries or continents. And hello, sweetheart. Oh, okay. You don't need me. Uh, okay, okay. Sorry about that.
That's okay.
Busy house. Just um. So that was one problem that was initially flagged, right? But there's also those yellow spheres halfway down the structure of this spike protein, right? And that's the other part of HIV, GP41, or another part of HIV called GP41. And they're essentially, they're two parts of a larger protein. And this is embedded in the what's now known worldwide as the furin cleavage site. And this this inclusion basically meant that this sarbecovirus clade was able to infect approximately 20, it was like 20 to 30 different tissue types relative more than the its contemporaneous known viruses. And so these two signals at the beginning were very concerning, but the epitope GP120 is a known amaloidogenic peptide that can cause the triggering of neurological disorders and the misregulation of prion protein as well. This is generally understood in the literature, and AIDS is associated with neurological disorders, and particularly in the end stages. It's called HANS, HIV-acquired neurological disorder.
It's, yeah, it's interesting. I met someone who had two vaccines, and that after the second one, he developed a brain and neurological issues. But I think what was particularly interesting about this case was, well, he tried a lot of different things, things that were recommended like NAC and Claritin for about 9 months or thereabouts. But then about 9 months, he found some side, and he ended up taking a HIV drug, and he actually said that he had a lot of improvement, significant improvement after he took a HIV drug. It cost him, $2,000. But before that, he was so disorientated he couldn't kind of navigate using the car. And now he's back to a reasonable level of functioning, but he still has, I guess, what you call brain fog. His brain is not as sharp as it used to be, and so he's still looking for mechanisms, I guess, to help his body recover. But it's an interesting link there, that what you're talking about between this COVID-19 and neurological disease and AIDS, or HIV, that it's all one big parcel, I guess, that people are trying to see it as separate, but it's almost a holistic thing. And also how we as a society move forward in terms of how do we treat this, or like we've got this problem now. How do
it gets much worse though in terms of the biochemistry. That was just the surface.
Oh, that's just
I'm afraid.
That
was okay. So that was the sort of initial, oh, something's not quite right in Kansas moment as we with seeing what we did see. And like I say, the work around amyloids and viruses has been a focus in the neuroscience domain for a number of years now. But the type of disorders, like I said, it was it generally leaned towards, well, you're going to have the neurodegenerative pattern at the end of life, right? If something doesn't take you out beforehand, heart disease, cancers, etc.
Accumulation of these prions, yeah.
And so the technical term would be like prion-like. I'm not sure I like prion-like. They're just prions, which means proteinaceous infectious particles, which means they're able to, like I say, start sticking together and form these cascades. Now, the orthodox thinking for much of my scientific career has been, well, you need the like-for-like peptide to cause the misfolding in the next one, and then the next one to cause the cascade to occur. And so this is, it's why we went to such extraordinary lengths around BSE. We thought we could remove these contaminants from the food chain and isolate it. And the concern was, well, what they found with the cows was that they were rendering brain and spinal cord and bone and feeding it back to the cows, and this was likely what caused the outbreak. And that's the story they tell us. I'm not so sure nowadays. But the start of BSE was very, very close to Porton Down, which is the UK's biowarfare research facilities.
Yeah, could be anything, really, couldn't it?
And as the technical know-how and scientific research has continued, we've come to understand that the formation of the prion peptide itself can be caused by what it's technically called cross-seeding amaloidogenic epitopes, right? So you don't need the whole protein, you just need certain segments of it. And once they're cleaved or digested, they're released, and then they can come into contact with susceptible proteins in the body and cause them to misfold. And then the process starts with the buildup of these amyloid.
Like a chain reaction, really. So
yeah.
you just need a primer, kind of like yogurt growing, essentially. You need
like,
to start
like what?
growing yogurt or something like that. You just get the starter, and it just
right, yeah.
It just I'm just trying to make it easier for people to understand the process within the body. Is like I've always thought a little bit suspicious that you can just have this basically tiny injection or you breathe in a tiny part, and all of a sudden, how does that one thing develop into this huge cascade of white clots, for example? And where does it all come from? The body itself has to be producing that somehow in some sort of cascade event. And then I think what they're seeing also that children are developing childhood dementia, and they've also noted that in the Pfizer document that the I1p32 gene deletion, which is associated with neurological, brain damage, is also one of the side effects. I mean, I guess this is the whole question: is it a side effect, or was it the effect? Was it, like, are we experiencing here a planned assault on humanity when you have this thing that's being actually unleashed that is actually an environmental toxin? And then where does it go? Does it go into the food supply? And they're also talking about injecting cattle and livestock with mRNA vaccines. Now, what are your thoughts on that? Do you think that will cause even more prion disease, more if people eat that, or is it?
So there's a couple of questions there. which the first one, let's deal with the first one before we get to
Yep.
the mRNA in cattle. So you were basically stating the premise that are we looking at a assault, a coordinated assault being made against the global population, and in looking at the aggregation of data right now, it's very difficult for me to exclude that possibility right now, only because of where the literal published science is leading us. And so, as the vaccines were being rolled out, there was it's an influential paper in the COVID field. It's from a Swedish group, Hammarström and Nystrom, which showed that there was a number of amyloidogenic peptide sequences studied across the spike protein of the virus in this instance. But in the preprint, they make a point of saying we should be very careful about using gene transfection as a method of trying to induce immunity because of the potential of expression of these amyloidogenic sequences.
Mm.
And this, the paper originally came out in 21 as a preprint, and then by the time the actual paper had been accepted, they'd had to remove any reference to vaccines, and this was indicative of the highly controlling environment in which the science was having to take place. And there was a whole range of measures being taken, and we're talking, the suspicion is, well, we're dealing with something that's come from this biowarfare medical countermeasures industry at minimum now, and we're looking at, well, okay, we're seeing fingerprints of what looked like so my friend Charles Rixey calls it de facto biowarfare. Whether it was deliberate or not, at this stage we don't know, but you just have to presume that, going by the evidence of what's emerging and what we know just about the molecular biology. And so I'm spending a lot of time trying to walk people through the what exposure to these peptides means, right? And I've got a monkey example that I can show quite elegantly what happens to the body and brain. But the view of well, what is it? Is it biowarfare? Is it something that nefarious? And it was kind of ambiguous in my mind up until very recently. And the same lab, Hammerstrom and Nystrom, in September, put out another preprint manuscript. I don't know if they'll ever get it into publication just because of how explosive it is. And I might just share screen again, and
Yep.
I'll bring that paper up. Of course, I will just shout out Luc Montagnier and Jean-Claude Perez, colleagues of mine now, and rest in peace, Jean-Claude, Luc, because Luc Montagnier was pivotal in bringing the HIV data to the public discussion. Okay?
Yeah.
So I'm just going through the slides here, but what I wanted to get to was this paper. So the title of the paper, if it's not fully clear on the screen, is SARS-CoV-2. So with this paper, so I'll read the title. It's informative. So SARS-CoV-2 spike amyloid fibrils specifically and selectively accelerates amyloid fibril formation of, and this is the critical part, human prion protein and the amyloid beta peptide. And this means that we have to take a very, very long, hard look at what this means with respect to biowarfare strategies and techniques, because prions have been a subject of biowarfare research for a long time. The problem was that you could aerosolize prion, the protein, and it's lethal in that respect. But the spreading is limited. And but as I mentioned earlier, as they focused on the weaponization and contraction, honing down the peptide sequences to their most essential disease-causing elements, I think that they've been focusing on how to find a way to get prion to spread person to person, not directly with, even though we consider prion disorder what's called TSEs, transmissible spongiform encephalopathies, meaning we know that if you come into contact with them, ingest them, there's a high chance that you'll develop the prion disease and you'll have a bad outcome, meaning invariably death, and a
Horrible death at that.
a horrible death. Yes, it's not pretty. So I'm just going to read out the part that I've got highlighted here. So they say, we here provide evidence of significant spike amyloid fibril seeded acceleration of amyloid formation of CJD-associated human prion protein using an in vitro conversion assay. So what that means is they're just, it's not in cell test systems yet. It's just, they're doing the molecular biochemistry in a manner in which they can detect change over time, right? And so you basically take this epitope sequence, and so they say we showed that the amyloid fibril formation of, in this case, Alzheimer-associated amyloid beta 142 was accelerated by spike amyloid fibrils of 7 different 20 amino acid long peptides. Spike 532 to 551, meaning just this 20 amino acid long sequence, was the most efficient at seeding human prion protein, whilst another segment, 601 to 620, was most effective at seeding amyloid beta. Now, if you just look at the data from their experiment, the fastest reaction that they have is with the conversion to the human prion, the scrapey form, meaning the diseased form of the prion protein in the data set, which is just this cluster down here, spike 532 seed. And so when we see a signal like this, on top of all the other data points that we've seen over the last 4 years, it's incumbent upon us to think, have these people, through malfeasance, perhaps it was just a research project gone awry at this stage. It's difficult to say for certain it's these groups, but I think as a scientist, I'm having to listen very carefully to the eugenicists and those people who dream of a far lower number of human beings on this planet. And if you wanted a method to do that, well, you're going to struggle through normal viral or bioweapon means, right? So you could go for the bacterial route, but there's likely we develop countermeasures in that respect. We can deal with them, and the onset of sickness, etc., has very typical presentations, whereas the spread of prion within a population would be very, very difficult to detect unless you are specifically looking for these misfolded proteins. And remember, we're in an area where it's more complex than just like for like conversion we now have to operate in a world of what's called cross-seeding
Yeah.
amaloidogenic peptide seeds, meaning these small epitopes.
And how would you even detect that? Do you want to close the screen?
Um,
Or you've got
so there's a couple more, but
yeah.
in the
How would you even detect that this seeding is happening within the population?
so the first thing that should be being done is that there needs to be mass surveillance of all types of tissues, from biopsies to bloods to um, brain tissue.
And that would probably only be gotten by people who are already sick, so it wouldn't represent the general population. So you
And
would
that
probably have a higher proportion of those that would have prions.
presumably. So and we have to remember that the presentation, because of the route of administration this time, which is that short chain catalyzing epitope has been directly gene transfected into people. And through this tissue agnostic, highly labile lipid nanoparticle, which means it can go all around the body and it can land in any tissue, and it'll go to areas where the metabolic demand is high. Cardiac,
Yep.
blood vessels, large blood vessels, and brain. And the and we're the picture is emerging that it's possible that cancers can be related to this type of misfolding process as well. So there's a whole suite of pathologies that wouldn't normally be associated with protein miss or prions in this instance, in the general sense, meaning the proteinaceous infectious particles,
And then they're
not.
going particularly fast too. Like we're seeing turbo cancer, turbo
Mm,
Alzheimer's disease.
mm,
So it's not like something we have a lot of time to get on top of. If,
right.
if if it is as bad as it is and it spreads throughout the population.
Mm.
Um,
And but so you asked the question: is it possible to detect right now? Well, there are ways, fairly novel ways of detecting this right now, and it's a little bit like PCR, where you can take a small seed from a biopsy and see if it amplifies and causes a buildup of prion under controlled conditions.
and you can imagine
And
that no one's going to want to be doing that after our health.
no. Of course, no one wants to do that now, and but, rightly so, considering what was done. And this is the important. There's so many points to cover, but it's such a complex area that I've I was going to mention this just a few minutes ago, but we have to keep going and trying to paint the picture down. But in order for you to have maximal impact with biological weapons, you need to have a not only the molecular understanding, you need to make sure that you have as much impact for as long as you can
Without being
across as broader swathe of the population as possible. And that's why, although it's not in the classic sense a binary weapon, because that implies two inert chemicals coming together to form the toxic agent. How SARS and the pandemic can be viewed is as a binary assault. So you had the virus released, okay? They had the mechanisms in place, and you have to understand that people who would have the knowledge of these weapon systems understood as much like a person who makes a guided missile or a nuclear weapon or whatever. They understand from the atomic level to the building out at the macro level what the effect is and how to use strategically. And they very obviously have understood that, okay, you need the first primer agent to go around the population. You then have the secondary come in which then bypasses immune barriers, where nature has learned to evolve to deal with these prion-like events. And this is a point I want to get to in a minute, but I would argue that they understood fundamentally how to line up everything, and that in totality is the weapons system.
Yeah.
You can't just look at the virus. You can't just look at the secondary delivery mechanism, which is this gene transfection assault. And you have to ask yourself, why were they so insistent on using gene transfection as the go-to remedy in this instance? They didn't have to do that, but they did. And what they've essentially done is that just from crude numbers, we know that approximately 13 billion mRNA type vaccines were delivered. That doesn't include the other kinds. And the 13 billion approximately into 4 billion people. And so essentially you've got your weapon to cover half of the human population in that 4 years. And if that was a weapons attack, they can't, now all they have to do is sit back, right? The job essentially has been done with respect to exposure. And, well, then you're going to say, well, I didn't take the vaccine. It doesn't matter in this instance, because we've got a real world example of what spread of prions looks like by looking at the American white-tailed deer population. And we can see over the last 20 years that there is an exponential rise across the continental United States, and now appearing in other countries. I want to say reindeer population in above the Arctic Circle in Europe, somewhere, Finland maybe, where this disease is happening. And the issue that we have here is that prions, again, because of their protease resistance, because of their ability to be persistent in the environment, and everyone is going to be shedding prion protein when particularly if you've come in and been infected with a heavy dose of the initial catalyst. Now, again, how good is their catalyst? I don't know. I would say this: if the premise and hypothesis is correct that this was a biowarfare attack, I would say they were probably pretty confident that it would do what it's supposed to, what it says on the tin, basically.
And we're all working from behind. Is that if they know what they're doing, we're always behind the 8 ball and trying to figure out what they're doing in terms of mitigating the harm and the damage from all that. And with the example with the deer, so you're essentially every deer is ending up being infected at some point.
Yes, and it's being infected because the prions essentially being secreted out through feces and urine. It gets taken up by root systems, and the deer eat the vegetation, and it continues the infective cycle. And there've been elegant demonstrations of that even just a month or so.
So if they wanted to speed up the process, I guess they could put the mRNA in meat and animal products. And
well, you would speed it up by
like if they
increasing the exposure.
They thought that people weren't dying fast enough, because obviously it's going to take
But
time.
you don't want people to die fast, right? Because that raises too many suspicions. So this is why you would choose something like a prion surreptitious disease mechanism, and one where it's attacking body and organ systems and manifesting in ways that are unpredictable. Now we know that now they're shaking out as cardiovascular type disease, or that's what they're being classed as in the all-cause mortality data of multiple countries. But we see that there's sustained increases in all-cause mortality across multiple countries. including where you are, including the UK, and the countries right now. I'm not sure how much longer we could trust any data coming out, as I'm sure that they would, as it's not an all-encompassing conspiracy, right? Because there are still parts that seem to be working, because you're getting data out that's pointing at that there's issues still ongoing, right? We can see that, but I don't know how much longer that's going to be the case. If it's correct that there's this prion. Well, so there's a phenomenon called the super prion hypothesis, and that's where I'll share my screen one more time.
So how long do you think before we get to a huge critical mass? Based on what's happened, say, with the deer?
20 years seems to be the number that the deer is showing us. I don't know. I mean, deer don't throng together by the thousands in stadia and cinemas and restaurants, and the route, particularly as it's been pushed past immune barriers, means that normal defenses are not at play anymore, and so it could make things worse.
And if you wanted, that when you unleash a weapon of some kind, what sort of antidote there might be to protect people, like
so this is something people ask me a lot, and let's start with this bit of data on the screen. So again, from this very efficient Swedish group, and this was published in 2014, and they published this paper, which is the prevalent human prion protein, and what they mean by 129M/V is methionine to valine substitution, which is what we would call a polymorphism in the protein. Is that archaeogenomic, meaning archaeology and extracting from genomes, signature of a superprion event in our past? And so it seems possible, with respect to the scientific literature and looking at our biology and the peptide presentation that we have, that there was this evolutionary bottleneck that occurred that caused this substitution to occur, such that you had a population collapse.
You making chocolate nuts?
No, ask Daddy.
Hello,
big boy.
Oh, hey.
How are you?
Good.
You good?
Yeah, I'm good.
Nice. What are you doing for today?
I'm going good. Just playing some games.
Video games? Yeah, like my kids.
Out you go.
Well, see ya.
Bye-bye. So I'll just read the section I've got highlighted on things. So for hitherto unexplained reasons, the majority of Europeans carry a missense mutation on one or both alleles of the prion protein gene, and hence express a variant of the PRP with a substitution for valine instead of methionine. In fact, the 12M9MV variant is very common in all populations except for Japanese, and Japanese is an isolated island nation that perhaps was insulated from the previous prion event that caused the population-wide adoption of this advantageous polymorphism. And so this whole premise in and of itself, I don't have to share the screen anymore.
Yeah.
You have to think about what strategic aims you're trying to achieve with the use of warfare and weapons like this. And one of those potentially could be, if you are of the you're part of an elite group, you think that there's just too many people around who they just know too much and they want too much, etc. And we've tried to convince them that the climate can't support them, etc. And so we may have to take active measures. And you can find the original authors recently of the Club of Rome limits to growth documents, basically saying that, well, what you would want is essentially not painless, but I guess, again, surreptitious ability to take out populations. But they also state do it in a meritocratic fashion, meaning everyone's up for selection through a potential evolutionary bottleneck, like would be required with respect to exposure to to a new disease-causing prion protein.
And essentially, that's like a survival of the fittest argument, isn't it? Their whole eugenics idea, where you create this situation where it's a test, essentially. Who can survive this prion assault will come out the other end, and those who don't will be the old would be taken out, the sick, those with potentially pre-existing conditions, except for maybe even the brightest ones amongst them who figure out what things to do with their own bodies in order to mitigate any damage and harm. So it's not so much that, and that's how you get the 500 million left. Is it's like in a lot of ways it's like passing through fire. That kind of, when you go back through the literature of human history, where they put it out in front of you, these ideas essentially of rites of passage, that we carry from, that this is the fair way, of putting this test out there. Just like the whole vaccine mandates were essentially a test, that it was always quite well known. They always put it out and seed it into the public that there were side effects, that you could potentially have heart issues and so forth. But now I think they never actually ever put out about the prions. They never said, you could get brain damage.
That was jumped on very early. Any mention of
they
prions.
They wouldn't want people to feel as though they could get brain damage, because heart, they can deal with brain damage. Maybe not. Even things like sexual dysfunction, they probably wouldn't want to put that out there. No one want to risk that, whereas heart issues, okay, I can deal with that. A lot of people felt that way. And I think even with the whole narrative that's going around, that did you get the vaccine, and whether you did or did not get COVID, like as though that's the biggest deal out there. Not that you got the vaccine and maybe you ended up with heart disease and all these other issues, and then passed it on to the rest of your family. Or did you really kill grandma, with by taking this mRNA technology that essentially transfected? I mean, how will the population feel being
they won't compute it. It just will not get through. I know this. It's too deep and harsh of a premise to take on board. You really have to want to be looking to see if this is see if it's real or not. And in a sense, like I say, if that's what's happened, the job is done, right? There's, like I say, there's mitigation strategies, right, when dealing with infective prions, but there's nothing in the armamentarium that will stop that process. You can just, like I say, slow it down. And in most instances, most people just wouldn't want to live anyway. So, it's almost a sort of losing battle from the beginning in trying to speak about these issues. For me, it matters because I want to know what's been done. And I think there's a importance in at least witnessing the events and at least speaking what it is that you know
Yeah.
to be
Not
at
to go
least a
down
credible.
quietly, I guess, that whole idea that we won't go down without a fight. And I mean, I think for some people, though, it would be interesting to know what your thoughts were in terms of mitigation strategies and treatments that, because obviously there's people spending a whole lot of money on things that potentially actually could be harmful to them. So there were, studies coming out that if you, broke down the spike protein, it actually could end up being more harmful,
more amaloidogenic.
which is it's kind of ironic, since you have a lot of people within, say, the freedom movement taking all these particular enzymes that they've been recommended, and they could actually be self-harming themselves and hastening their own demise. So from your experience, and your research, what would you suggest, like, to people? Are we looking at water fasting, intermittent water fasting? I've heard various diet supplements. I think we mentioned it earlier before, aspirin.
So I take the view that if you want to be able to try it and see if it might turn you around, maybe you are in like the long hauler type conditions that we're seeing, I would argue is primarily a consequence of this protein misfolding disease mechanisms and immune dysregulation. And so I think you should have access to and try whatever it is that you think might work for you. I'm not going to say don't try. I'm trying right now with colleagues in Germany to look at methods to clear the amaloidogenic microclots from the blood and the plasma. And we see some quite elegant turnarounds with people who have been essentially wheelchair bound, and people who have been harmed by both the virus and the vaccine in this instance. But is it a feasible long-term strategy to be having your blood cleaned every 6 months in perpetuity? I don't know. But that for me, that's that in terms of real concrete steps, I would be checking to see, or going down that pathway and trying to see, like I say, when people are looking in their blood on the dark field and they're thinking they're seeing nanobots, etc. You're not. You're looking at amyloidogenic clots under the scope. That's what they're looking at, and you need to try and remove those.
So when you're talking about blood cleansing, is that EDTMA or something like that? That
the collating agents. But yeah, look, these are all approaches that I would take were one trying to stave off a assault of this kind. Again, I can't guarantee in this fashion, particularly in an environment where everything now could essentially just be considered contaminated, unless you're able to sequester yourself far away from large groups, even moderate population centers now, because literally everyone in the first world has had a exposure and multiple exposures now. And so it's, you asked me earlier, like, what would be the countermeasures that those who planned this were in? Maybe they didn't have any. Maybe they're such believers in their own ideology that they believe that they've got to go through this
or maybe
process
that's genetics to us.
Well, maybe, yeah.
Because after all those all those genetic collection agencies where people were sending in their DNA, and we've had that for very many years, where you had the Guthrie cards where every newborn baby had their bloods taken and stored, and it's very difficult actually to get those Guthrie cards back if you want them. So they know they had the full picture initially if if you wanted to, if your aim was essentially to cause a mass extinction event, and you had all the data points, you would know what you were
Yeah, and
targeting.
if you have foreknowledge of what's going to happen, then you can just position yourself in such a way that you've limited your exposure. And from the clinical data prior to, well, in the prion space, it was that most people, even if you get the spontaneous CJD, it's in your 60s, etc., and you've had time to live somewhat, hopefully useful life.
Yeah. So what were we saying? Yeah. So like things like NAD plus and glutathione. I think this guy was talking to you about. He used 4 or 5 rounds of that because he had such a huge vaccine reaction. But it's actually, I don't know. With Dr. Richard Fleming's approach to it, I know you've interviewed with him. He seems to believe that the immune system will be able to mount a response to this. He's got more of a immunothrombotic idea about what's happening within the body, as opposed to the prion side. I think when I did discuss it with him, he talked about niacin in dealing with amyloids. I'm not sure what other research there is, but potentially if we live long enough, we will have to find, we hope that we either evolve or...
But from a
sort of
species perspective, the way through is that the human population has to come up against that evolutionary pressure and adapt and overcome. That's the long-term solution. People who, again, when you're dealing with a biological toxin in the form of prions, and you're just essentially surrounded by it all the time. Again, anything that you're going to try and do is just stave off the inevitable. And I don't want to dissuade people from taking any measure that they think that they would like to explore. A lot of people are contacting me about methylene blue at the moment because it's one of those agents which in vitro will disaggregate fibrils. None of this has been shown to confer any sort of long-term benefit in clinical cases, and this is, there's limits to what we can and can't do, and nature's going to find the solution if that's what it is that they've done. I pray to God that's not what they've done, but it doesn't look good right now, and what comes next could be way worse than what happened with COVID, because the technology is here now with respect to being able to identify the prion agents a little like PCR. And so they could, in theory, whilst then, if you want a supporting data point, in the next 7 years, there's going to be basically 50% increase in drugs for treating prion disorders. These are projections, sales projections.
So they've already made these drugs.
No. These aren't drugs to stop the disease process. It's to mitigate the symptoms of the disease. And so the growth is in antipsychotics, antidepressants, and various other means of clamping the nervous system to a particular state.
And they could even target the natural field by using the magic mushrooms, those people who may not want to take the pharmaceutical drugs, you'll have that in the and not hemp, what was it? Marijuana, the whole medicinal marijuana. It's interesting how that's all being promoted, you know,
Mhm.
that you have this rise of mental health issues, and so people who want the pharmaceutical side have got their, you know, entrees and main meal, and then you've also got those who refuse to take pharmaceuticals, who are encouraged to take the natural route of various herbs and supplements that have been also synthesized within a lab. And the pharmaceuticals wins both ways.
Yep. Yeah, you get it.
You know, it's kind of checkmate, really, in a way, except for those who can. And I think this is where, you know, for those of us who are in this, it's about claiming accountability here and making sure that this is documented. This is, you know, if it's all that we do, like if this is where we go down, at least we
well, it's to hold people to account. That's the reason for doing this. And you, in the time, if we're against a ticking clock, to make sure that if you know, if there's infrastructure left over, that there's documents written down for these people. Don't do that again. It's it's painful when people find out that we've potentially caused something of this magnitude. That's what needs to happen. And I would say with the current state of the art with the diagnostics and molecular biology, we could do we could go through these institutes and these individuals who we know would have been at least front side to the public, who perhaps orchestrated something like this, and to hold these institutes to account that way. And that's kind of what I was hoping. And again, you can just, all we can do right now is just hope that the worst case scenario, it hasn't happened in this instance. But I would, and if people are listening to this, what we experienced could have been used in that fashion. That's how vulnerable you are right now. And so you've got to move away from these centralized systems where they were able to, again, synchronize everything to get this maximum exposure the way they did. My wife's telling me that I've got to dip.
That's right. We can wind it up.
So yeah, on that thought, I would just say, yeah, it's still we can hold these people to account potentially if people want to help, and you know, that means petitioning politicians. There is the scientific basis, and we can move forward that way in terms of concrete steps, as well as looking towards trying to again mitigate. And don't listen to when they tell you you can't have future generations, etc. Have children. That's the best way forward.
Because that's the evolution, isn't it, that we after. Anyway, thanks everyone for watching today. Thank you, Dr. Kevin McCann, for all your time and what you've shared with us. I know it's incredibly hard. I will put links to your, I think it's your website, isn't it? You know,
yes. I mean, look, I've had to wage a guerrilla campaign for like the last 4 years. So this is generally a more, how should we say, professional take on what it is that I do. I have to survive, basically. So
yes,
you know,
yes. I'll put a link to your website so that those people here who want to support your work. We will be hopeful that you know, life's a long journey. We can hope that people can innovate, and who knows what the future may hold. I know it's kind of a bit of a dark episode, but it really does sort of highlight the seriousness of what has happened here, and at all. Anyway, thank you so much.
You're welcome. I'll hopefully speak to you again soon. Hopefully without so much family drama going on. So
That's okay.
okay. All right. Take care, Jean.
See you. Bye.
Bye bye.